So I want to welcome you here. This is a special lecture to me. This is in dedication of a patient of mine and Dr. Antonio Menos, Mr. Paul Whaling, who was really an amazing guy who had advanced laryngeal cancer. He was had a very strong background in electrical engineering, was an IT person who managed over 100 people, just a very devoted father and even grandfather. He actually had another side gig where he was a professional musician, and he actually grew up playing guitar and was in many, many smoky bars and different hangouts when he was young. And interestingly enough, he had never smoked a day in his life, had laryngeal cancer that was P16 negative. So the thought was, all these years of secondhand smoke led to this laryngeal cancer that was locally advanced. And his passing in 2011 really kind of had myself and Antonio look at each other and say, can we do something that's a little bit different? Can we start to think a little bit out of the box in what we're doing here? And led to some important clinical trials that are being quoted right now around the world. And in fact, I just got off a phone call with the cooperative group on a design of a trial similar to ours, where we used a PARP inhibitor for heavy smoker or for DNA damage repair or defect head and neck cancers with dual biologic approaches and taking the cisplatin out of it. And so based on what we did here, it's really making a difference in terms of establishing clinical trials around the country and around the world using DNA damage repair inhibitors. And Karen is here, his lovely wife, who looks like she could be his daughter anyway, and their daughter Beth. Karen is a cancer survivor as well and has been treated here at the University of Colorado under the GI group's auspices. And we're so honored to have them here. They've meant a lot to us, and we've developed a long-standing relationship for over many, many years. And I'm very proud to begin by having, for the first lecture here, Dr. Joanne Weidhaas, who not only is a friend and a close colleague, but really is, again, someone, when I try to think of people who are thinking out of the box, because that's really what we need to do to become better clinicians in head and neck cancer, I thought of Joanna in terms of some of the important work she's doing at trying to find ways to predict response to radiation, radiogenomics. And her work is really centered around germline mutations as cancer biomarkers. She is a full professor at UCLA and head of the translational program there, has a very extensive background. She did her undergraduate, I think, at Yale and then Tufts Medical School, and then did her Holman Pathway and Radiation Oncology training at Memorial Sloan Kettering Cancer Center, and then went back to Yale, and then more recently, over the last 4 or 5 years, gravitated out to UCLA. She's one of these people that I just wish I was as smart as. She started her own biotech company, as well as being this incredible translational researcher and a person who has a lot of passion and excitement. She's been R01 funded successfully in the past. She has a very important impact, high impact publications. I was honored to be on one with her, looking at the use of KRAS mutations as a way of predicting response in head and neck cancer trials of cetuximab and radiation. So I'm really honored to have Joanne here as our first Paul Whaling memorial speaker. And without further ado, I'm going to have her. I apologize to her about the weather here. She's coming from Santa Monica, and so to us this is a balmy day for us. But she I think is already missing Santa Monica. But we're fortunate enough to have her here. So
Thank you.
I'll close that there, and then yours should be open.
Awesome.
There you go.
Okay. Check that out. Push the button at my booth. That's cool. I feel like a superhero. Thank you so much for the very nice introduction. Thanks for coming, everyone, today. Could I ask you to start? How many people in the audience are physicians? Can you raise your hand? Small subset. Okay, good. So I made this my attempt to bridge together the science and the clinical implications, and for lay people as well. So we'll see how it goes. So as David mentioned, I do have a company that I co-founded. He failed to mention I took a slight detour and went to business school up in Northern California, and a nonprofit that focuses on women's health. And that's related a bit to the work we're doing that I'll talk about. So I'm going to talk about microRNA germline genetics in cancer. And I'm not following microRNA profiles I'm really just talking about the DNA you show up with, that people have differences, and those differences affect microRNAs. So just to keep everyone on the same page, I'm going to start with kind of an overview of cancer. Just some basic facts that even if you work on cancer, you may not really know those numbers. It's good to keep in perspective. I'm going to explain what microRNAs are, why they matter, and why we think baseline genetic differences in microRNAs really can help us better personalize cancer treatment. And I'm going to do this by kind of explaining the findings we find in patients and clinical trials back to cell lines and basic science models, and how we're trying to, biologically model our findings about biomarkers we know are clinically really important. And that's a little challenging, but I'll tell you about that. Simple facts about cancer. Cancer doctors aren't allowed to answer. How common is it? Do we think there are 2 million cases in the US a year, 1 million cases, 500,000? Someone shout out an answer. Come on. 2 million, 1 million, 500,000. Some non-cancer doctor, just say something. Okay. It's actually closer to 2, so that's in the US alone. 1.7 million, not bad. You got a B+ on that. In the US alone, there's over 6 million cases a year in the world, so a lot of people. That's actually a diagnosis of cancer every 18 seconds in the United States. So cancer is a really common problem. Second question, who gets cancer? How many women and how many men? Do you think it's 1 in 25 people, 1 in 10 people, 1 in 2 people? You're cheating. You're not allowed to say. It's really common. David, someone get some tape. David can't answer. It's really common. Almost half the people in this room will have cancer. Sink that in for a second. Half the people. And if I ask you who you are, everyone is going to say, it's not me, it's that person over there, because you're healthy. You're not smoking. You're doing all the right things. No one thinks they're going to get cancer. And I have a lot of really healthy cancer people that look just like the people in this audience. So this is why you need to know this. It's no one's fault if they get cancer. Half of you are going to get it. So not to be horrifying, but I think we have some answers. Why do people get cancer? Right? That's the million-dollar question. Why will half of the people in this room get cancer? I never had a cancer patient that knows why they got cancer. The most common thing is age. Okay, so you're pretty clear until you're 55, which is getting close, quick. 3/4 of cancer happen just from aging. Exposures. Certainly some exposures affect some people. There's viral association certainly with liver cancers. Smoking is a risk, although only 10% of smokers get lung cancer. So it's not the same. It's not like everyone that does something will have something bad happen. But the number one risk of cancer is actually a family history of cancer. Family history of cancer. So what does that say? That you already have that risk in your DNA. The problem is that we can't tell you that, most of the people, because we only have markers that explain about 2% of family cancers. BRCA genes, those very rare other genes like BRCA, because they were identified in people that get cancer in their 20s and 30s. And guess what? That's not when 3/4 of people get cancer. So why does everyone else get cancer? So, training a memorial, seeing all my healthy cancer patients, this was clearly a question that I felt needed clarification. Another thing, more cancer facts. How good are we at curing cancer? If you're diagnosed with cancer, what is the chance you'll be cured just as for everyone? Is it 80%, 50%, or 20%? Non-David? Anyone? Come on, don't be afraid. It's actually second leading cause of death in the US, cancer. Heart disease is first, and they kind of go back and forth. So it's not great. A third of people will die of a cancer diagnosis. Of course, this varies enormously by the type of cancer you have. Some cancers we are very good at curing. This is 5-year survival. Notice melanoma can fail later. These are women, these are men. Some cancers we do very poorly. Pancreatic cancer, very hard to treat. And a lot of this depends how early we pick it up, but even early cancers that are in certain organs, we still do poorly at. But I'll tell you, if you're someone and you have one of these cancers, no matter what the odds are, you say, I'm going to be the 5% that lives. I'm going to be the 80% that lives, because you're smart, you You have great doctors like you can get here. You will do all the right things. And the truth is, as the doctors here can tell you, we have patients do all the right things. They're smart, wonderful people, and they still can fail. So we need better ways to personalize cancer treatment. We need to know what to do to cure you individually when you get cancer. And I'll tell you where our challenges have been and why I think we made a lot of progress, and there's a lot of hope for that that's coming up. So there are challenges. How do we find the answers? Where is the answer to who's getting cancer and how can we cure you? Is it what you're born with? I did tell you, genetics, #1 risk for cancer. So there's something with what you're born with. Could we use this to know that? Or much of what we've done in cancer is say, let's just look at your tumor. Whoever you are, wherever you came from, you made a tumor. We're going to look really hard at that tumor, and we're going to find an answer. And all your tumors are kind of different, but we'll find something that tells us how to treat you. Or is it just the luck of some things work? We just need a better treatment that works for everybody, right? It could be any of these things. I'm going to try to convince you that they're related, that I could tell you things you have now that you're born with that will tell me how to treat you, and that actually will have shaped your tumor. And so it all makes sense, all our findings in tumors, but it's based on something you start with. And knowing that information, I will know how to treat you. So I'm going to try to convince you of that. And it's important I have a science slide to explain where I think the answers are. Now, what we based all our markers on are an original understanding of DNA, which we have DNA, we make messenger RNA, and we make proteins. This is really old findings, literally from the 50s and 60s. But that has been all of cancer biology has looked, and all existing biomarkers are in this part of the DNA. But that's a tiny sliver of our DNA, 2% of all our DNA. And when they sequenced the genome, they said, geez, we got a lot of junk in our DNA. People just have a lot of junk. They got a lot of DNA, but we only use 2%. We're not very sophisticated. But of course, as is the case in science, the truth is we just didn't understand. We just didn't have the understanding. And I always think biology is kind of a beautiful thing. There really is. We just don't understand the explanation. There is an explanation. Turns out that we use tons of our DNA. It just doesn't turn into proteins. And it's called the non-coding RNA, or DNA, sorry, and it makes these non-coding RNAs. And these were really recently discovered. And the way that they work in the first class, which I focus on, are microRNAs. They're a tiny little piece of RNA, like tiny, tiny compared to a protein. They used to wash it away in the test tube. They thought it was broken down. They thought it didn't matter. Turns out that they are huge, important global regulators of our whole self. They circulate. They tell you if you're infected, if you had a heart attack, if you're getting radiation or chemo. They coordinate your response as a human being to that stress. That's what microRNAs do. And they actually primarily work, so they come from part of the DNA we've never looked in, and they work through the messenger RNAs in a part that we haven't ever really looked at because we didn't think it mattered, the poly A tail. And they organize these whole stress response programs. That's what they do. And what's cool is that microRNAs are relatively incredibly new. We didn't know about them until the late 90s. And now if you go to a non-coding RNA meeting, they'll say, our paradigm's been upside down since the 50s. The proteins don't matter. It's the upstream RNAs. And that's why we have not found all the answers we want. I'm going to try to convince you somewhat that that's true today. And it's 80% of our DNA is active all the time, that we can't live without. It's incredibly important. So makes you very hopeful that even though we've had some blockbuster biomarkers in this 2%, maybe a dozen. Think of how many we could get out of 80% to help us treat. So that's what I'll try to show you. Okay, so a little more science. I'm just going to explain globally what microRNAs do for the science people. You'll appreciate it more. We've known since the beginning that microRNAs are tissue specific. They found they're important in development. So from the time you're fertilized as an embryo, the microRNAs for the lung will make a lung, will make a liver, will make a brain. So that's where they're first discovered. There's about 2,500 microRNAs in people. They target probably all the messenger RNAs. Really important development. As I said, tissues are unique. What we know is if you look in a diseased tissue, a cancer, their microRNAs have all changed. So they're important. And And so when we started out looking at this, we said, but how do we use them? You know, you have them to develop to make a lung, and they end up screwed up in a cancer. But do you use them day to day? Why do we still have them? And actually, some of the first work that we did was looking at that. And it was the first evidence that microRNAs changed immediately with stress. They organize that stress response. So I should tell you, so for the non-science people, these are different microRNA expressions. Green means they go lower compared to where they start. Red means they go up. And these are a normal lung line, a cancer lung line. What you see is in lung tissue, microRNAs change the same way, cancer or normal. This was within 2 hours. We now know within 15 minutes microRNAs change. And they dictate a very coordinated response. This is stress, damage, stop cell cycle, bring in DNA repair. They pull it together. They regulate about 500 messenger RNAs, so they can stop and start whole programs. Now the truth is they've shown this if someone has a heart attack, microRNAs, if you're infected, if you're septic, anything you do, microRNAs are kind of the first responders. So that's all I want you to take away from this. This is just a little deep science talk for radiation. We find that there's ways we still barely understand about microRNAs. The miR34 microRNA, really important in the radiation response. It was always thought microRNAs were made and then could be they could be upregulated from the DNA like proteins. It turns out there are mechanisms. If you have miR34 floating around, you can actually add a phosphate and activate it. It's never been known to be happening except in proteins. So that's just kind of a science thing. So there is much we don't understand in microRNA biology, but this is critical in the radiation response. It's a critical early responder, so important that it sits around waiting to be activated. It's activated in 15 minutes. So I won't go into that. As I mentioned, microRNAs are always abnormal in cancer, and this is relevant to some of the findings we have. So we know that there's certain cancers have very specific microRNA abnormalities. We know some microRNAs are oncogenes, so they're up. So miR21 is in here somewhere. It's almost universally up, which is red. LET7s are tumor suppressors. They're almost universally down. Other ones, it's a hypoxic environment, a tumor, so they're down or up. And with that, we found we could look at a tumor in the microRNAs. We're focusing on the tumor, and we say, oh, this predicts something. Predicts outcome, predicts poor survival. It tells us a tumor might be ugly, might be aggressive. And there's been a lot of work to try to give back microRNAs that are missing or knock down microRNAs that are too much. It turns out they're so powerful microRNAs that if you try to give someone microRNAs, they have huge inflammatory responses, and the companies have shut. They're too important in the systemic stress response. We can't do it, even though we know they're important in tumors. So what does that mean for us? The question is, and this is where we have focused, is how do we get, where are microRNAs in this? How do they get from where we start in your tumors, and how do we choose your therapy based on them? And, I'll talk about where we started trying to investigate this. We just want to know, why are microRNAs abnormal in lung cancer? Why is LET7 low and KRAS high? And I'll tell you, like everyone, we thought it was going to be about the tumor, but we found it was about the patient, so I'm going to tell you about that. So we looked, how do you make a microRNA? Let's look in lung tumors. And as I mentioned, the microRNA comes from your DNA, you make your microRNA, and then it hits its important target, which in this case was KRAS. We looked at tumors, we said, what happened? Did the tumor mutate the microRNA so it couldn't make it? Did it not let the microRNA bind to KRAS, or KRAS went up? Makes sense. I mean, that's how we look at it. The tumor's evil, it's done something, it screwed up our microRNAs, which are critical. And what happened was we found that we never saw microRNAs were mutated in cancer. That's not what happened. The cancer didn't do it. So it is not the tumor that causes the microRNA problems. So we said, all right, what does that mean? And we did this huge sequencing. That was actually a grant we got to do that, and someone published the results before we got the money. And we're like, wow, that is good, because we didn't waste the money, but where do we look? How do we know why microRNAs are abnormal? So we started a huge sequencing project of paired normal and tumor tissues. And we looked at the places this microRNA targeted KRAS, important oncogene. And what you see, so here's the messenger RNA, the KRAS protein. Everyone's heard about that. We didn't look at that. We looked at where the microRNA binds. And there's a lot of places LET7 binds. And what we found was something that we have in all our tissues. That's in about 10% of people. So 10% of the people in this audience have this. It's something you're born with a different KRAS, all right? A different way for your microRNA to communicate with your KRAS. And, 12% are Caucasian, 6% of people in the world. The first study we found was that it was higher in lung cancer patients. And it seems like if you walk around with this, you are one of those regular people. You look normal, you seem normal, who has a slightly increased risk of certain cancers. And it's really not the same as a tumor protein KRAS mutation. It's so fundamentally different, and I'll talk about that a little. So that's about it being in the patient. So this is something you start with that predicts cancer risk. So that was our first step. This has been really widely studied. The great thing about having a germline marker is I can pick it up in a cheek swab. I can pick it up in blood. We had a huge study in the Netherlands where they used toenail clippings. It's crazy, but they collect toenail clippings from everyone when they hit 50. They have an incredibly I know, gross. Apparently really hard to get DNA, but there's DNA in your toenail clippings. I wouldn't want the job, but it was a great resource. And what we found was, if you're someone with KRAS variant, you were to increase for certain cancers, increased risk, non-small cell lung, ovarian, triple-negative breast particularly. If you're from a family with a strong family history, it's in more of those patients. These are not huge risks, but 150% increased risk is high enough. And what we really know now, actually, is that this is a huge risk for women. And I'm not going to talk about this today, but the risk happens with estrogen withdrawal at menopause. That's the risk, taking away estrogen for these people. So, and we have a lot of biology going on understanding that. But, so we went back to the bench and we said, all right, we need to kind of understand how this works because we find it in clinical trials, but I'm a scientist at heart, and the truth is it's hard to move things forward without some scientific understanding. So I'll tell you a little bit about some very tedious biological experiments where in the day before CRISPR, or it's Cas9 for human cells, we created perfectly matched lines with or without the KRAS variant. It was the only way to do it. You can look at cancer cell lines that have it, and it's in the NCI 60 and like 10% of the lines, but they've been growing for 40 years, and we wanted a perfect match. So we created a panel of KRAS variant lines, and very tedious at the time to do it, and confirmed it was the only difference between the lines. But we have now an array of both cancer lines as well as some normal breast cell lines. So we could say, if this line has a KRAS variant, is it different? Is there anything different about it? And we right away saw that it was quite different. So these are two isogenic lines. So these are normal breast cell lines. This is what it looks like normally. Grows happily in its tissue culture disc, which of course is a fake environment. But in our cell lines where we had the KRAS variant, they shifted and were very mesenchymal. Really obvious to the naked eye. And, this project just got accepted as in a publication, but literally took 10 years to publish because people said, that's impossible. You can't put in a single point mutation. And so, my poor postdocs. So we've done years of work, 10 years of work, and I'll show you the continuation of it, but they really had a strong mesenchymal phenotype. They wouldn't form a Rossini. Genotypically, they were clearly mesenchymal. They had low ECAT here, and these are just our standard mesenchymal epithelial versus mesenchymal, very high fibronectin. Snail was high, as was Zeb. Like all the things, they were just mesenchymal, and there were two independent isolates. Just from this mutation, we did whole sequencing. We did, everything to prove there was nothing else different as technology improved, but we didn't really know why. Like, how did KRAS's change lead to all this? So I'll tell you what we learned. We went back to the bench. Again, this is just based on something you're born with. These are normal epithelial lines. This is something people walk around with. So we went back to clinic, and this was all going on in parallel. And we said, okay, if you're a KRAS person, you start different, it seems. Does your tumor look different? Does it matter? Does it take you through that continuum? And we found it did. So actually, if you look at several groups, looked at KRAS variant tumors, tumors in people with the KRAS variant, breast tumors, ovarian tumors, lung tumors, head and neck tumors, and every time they found, they really have significantly low LET7. It's always low. If you're a KRAS variant person and you make a tumor, your tumor looks like that. So, but even more, if we looked at the tumor gene expression, and this was done in triple negative breast, where there's a lot of gene expression done, These are KRAS variant tumors. These are triple negative tumors without it. They look the same. They use the same genetic pathway. And that's really different than how we view cancer. First off, even people with BRCA mutations, their tumors don't look the same because it's a random hit. They got a lot of mutations, and their tumors are all diverse. Here we have something you walk around with. You make a tumor, and be it a breast tumor, an ovarian tumor, a lung tumor, or a colon tumor, that tumor goes down a common path. So that's a paradigm shift in cancer, which to me just means uphill battle, paradigm shift. That's been my life. So they really have very unique gene expression, and it's a KRAS-addicted signature. They don't have tumor-acquired KRAS mutations. That's rare in KRAS-variant patients. They kind of don't need it. The pathway's already on. But they do have a KRAS-addicted signature. And they have very high AKT expression. So that's one of the important things we found. We asked, is it just triple-negative tumors? I already told you microRNA expression is common across tumor types, and the answer is no. We looked at ovarian tumors. We saw exactly the same KRAS-addicted signature. They're very resistant to platinum, and that's true in breast cancer and head and neck cancer and colon cancer, in ovarian cancer, where it's very well defined. So you're a person that starts, you make a tumor, it looks the same. So how do we use information? Here we've been saying, you know, our model has been look at the tumor and figure out how to treat. So theoretically, we should be able to do the same thing. And so that's what we've been doing. So at this point, there have been thousands and thousands and thousands of people tested for the KRAS variant, so the single DNA change, and to see if it impacts their treatment. And so we have a lot of studies, many independent of this, but there are certain drugs that are fantastic if you have the KRAS variant. There's a lot of studies in non-small cell lung. These were the Battle 1 and 2 trials, as well as an independent trial. A lot of work in colon, this head and neck trial I'm going to talk about a little bit more in depth. A lung trial, which we haven't published yet, for cetuximab, great drug, improved overall survival. So these aren't just, a 1.1 odds ratio. This is 6-fold increase in overall survival if you get that right drug, right? Cisplatin, they're always resistant. Erlotinib is a terrible drug for them. Vandetanib, which is part of angiogenesis pathway, terrible drug for them. So why? So here you're born with it, you make a tumor, and then you have really different responses to drugs. And is it just the tumor? Is it really your whole systemic response? I told you microRNAs are really important in your systemic response. So I'm going to tell you what we learned in a radiation trial about the KRAS variant and how it really showed us that these predict your normal whole body response, and how now we've applied that in other places. Oh, sorry, I blocked out my own writing. So this is actually taking something I pick up in a cheek swab and choosing a drug for you. And I could do that today because that marker doesn't change. I could say, give me a cheek swab. If you ever get cancer, take that and don't take that. And that someday is where we need to be in personalized medicine. Or this is safe and this is super not safe. And we have a lot of toxicity info now, which I'll show you. So I'm going to talk about this randomized trial, which David referred to. As we do in many of our clinical trial, the science looks awesome. We do a phase 1, it looks great. We pick a cancer type. Phase 2, it looks good. We go to phase 3, randomized trial. Half of you get this treatment. You all have the same cancer. Half get this new improved with the drug that looked really good. We think it's going to be better. Unfortunately, probably 80% of the phase 3 trials, 75% fail. They don't show the benefit. And it's not that there are not people that didn't benefit, it's we can't identify them. There are people that did better with that drug, but no one ever gets it because it wasn't approved. And then pharma just puts it on a shelf because they don't know who it worked for. And they must have been in the phase 2 because it looked really good, but they weren't in the phase 3. They don't have enough of them. So this is one of those many trials, and unfortunately, we've had quite a few in radiation where we're adding some new agent to radiation that looks so good, and it fails. So this was a phase 3 trial, head and neck cancer. People got standard radiation cisplatin or radiation cisplatin cetuximab. We worked with NRG. It took several years, but we did get the samples, which was great. And we've got a couple phase 3s from them. And other people have shown cetuximab is a great drug for people with our KRAS mutation. Terrible if you have a tumor KRAS mutation. Great if you have this inherited KRAS mutation. And this is what we saw. So I'll talk the non-clinicians through this. So this is progression-free survival, and these are years, and this is overall survival, and these are years. And what you see, the dotted lines are people without the KRAS variant. They did about the same, right? They kind of came along. Eventually, you started 100%, but over time, people die of disease, progress or die of disease. The solid lines are the people with the KRAS variant. Blue means they just got radiation cisplatin. Red means they got cetuximab. So what you see, the solid lines, KRAS variant patients really separated on this trial, right? It was a fantastic thing for them for progression-free survival that held them for about a year. Note, it's 8 cycles. They got 8 weeks of cetuximab, then it was done, okay? Overall survival, it kept them alive significantly for 2-year benefit for overall survival from 8 weeks of cetuximab. Compared to the people without the KRAS variant, there is no difference. So this is the group. They respond, right? And what's interesting, there are a couple very interesting things we learned from this. One, that the P16, the HPV positive patients, they had a fantastic response. And I'll just show you that. Where you see this is overall survival. People with the KRAS variant that were HPV positive with cetuximab, they were cured. 95% 5-year survival from 8 weeks of cetuximab. Fantastic. But if they didn't get cetuximab, they did terribly. That's 70% 5-year survival. That's bad for an HPV positive patient. And when we did it really carefully by subgroup, we actually saw at 2 years, we see a significant difference, 95% versus 60% survival if you don't get cetuximab. So if you're out there with a KRAS variant, right now cetuximab is not approved for your head and neck cancer. So you're not going to get it. And your survival is 60% versus 95%. So this is the problem, not knowing and putting in those markers, right? The good news is, and I'll tell you, but Lilly is finally funding a prospective trial of just KRAS patients for cetuximab. But it's taken 8 years to make that happen. So what we found out was, why does cetuximab work? And this is what's relevant, is that it wasn't just about the tumor, but it was how that patient responded. And what happened was we found they had really different baseline immune systems. So KRAS variant patients, you're walking around, you're tipped a little way, you have really low natural killer cells. And one day in evolution, when people were dying of the plague from inflammation and KRAS variant patients didn't, it was a really good thing to have. But when you're being treated with radiation and you want to stimulate your immune system, it's not good. They don't respond, and they have very elevated TGF-beta. So we could see that in that phase 3 trial, okay? We actually really also saw that when they failed, cetuximab protects people from dying of metastatic disease. So it keeps you from growing a tumor somewhere else. So that all makes sense. It's your immune system. Radiation, and some people simulate that. If you're a KRAS variant patient, it doesn't cut it. And you need cetuximab to do it. So that's our theory. Just back to the biology for a minute, since we have a lot of biologists. So we see elevated TGF-beta in cancer patients. We have this normal tissue model we're trying to model. Do we see that? So first, looking at the tumor, microRNAs, very abnormal in the cell line model. Significantly altered. That's 7's low, but lots of them. MIR200 is incredibly low. It explains mesenchymal phenotype. We also see very high AKT, like we saw in tumors. Very consistent in the cell line model. And importantly, just in these cell lines, we saw incredibly high TGF-beta. Huge. TGF-beta 1, 2, and 3. Super elevated just from putting in the KRAS variant in the cell line. So people work with cell lines all the time. You pick one cell line as your model. You might be looking at really fundamental different biology. They're both breast. They're triple negative. They look the same. There's underpinnings in genetics that make us all incredibly different, right? Anyway, so we could show it's TGF-beta, it's AKT pathway, which totally all makes sense. And we could, if you inhibit TGF-beta, you actually could reverse some of the mesenchymal phenotype. And so that is our proposed model for KRAS variant people. You know, they have their crazy biology. They have this single mutation. It changes microRNAs, and they secrete all types of different factors. In a stress condition, I mean, tissue culture is a stress. But if you're going down a path to cancer, you end up that same way. You have very high TGF-beta. You have modified the milieu. So that's going to impact how you respond to treatment. It just is. So these are underpinnings that are good to know. So I'm going to just say this is where I've hopefully gotten you to at least believe it's possible, and I'm going to give you a couple more examples where we've applied this type of mutation, which is kind of further proving the paradigm. I believe we walk around with different baseline biology, and we can identify it. I could test you all today and tell you who has a KRAS variant. That's the first example. Takes me an hour. I'll do it for free. I'm such a bad business person, but I will. That's why I will always just need to run a nonprofit. The truth is, if it's a KRAS variant person, there are different triggers for you from cancer. You're doing what your doctor said, don't take hormone replacement therapy, and now you have a triple negative breast cancer. Shit. But you're doing what everyone said. You live a super healthy life. And you're not fat, because actually fat protects KRAS variant patients from cancer. So you've lost a bunch of weight, you've stopped your hormone, and now you have cancer. Those are my healthy cancer patients. You did everything right. So if you're a KRAS person, when you're pushed, we do a trigger, you're triggered, you didn't know what it was, you develop a tumor that has common biology, and you as a person have created a unique, well, like other KRAS variant people world. Now we can select treatment for you because we know, and we don't need, we can always correlate with a tumor, but we can tell you in that cheek swab, this is you and this is what you should get. And that is our hope for this class of mutations. This is the potential. So now I'm going to tell you a few other paradigms where I think that we're seeing that. And it's not just the tumor, because I really believe it's more about a person's systemic response to stress, where they are at baseline and how they respond. And I'm going to try to prove that to you from some of the other applications we had. So we spent years. Of course, KRAS, it is not one single point mutation in all of our DNA. There are a lot of other mutations like it. They are in parts of the DNA that no one has looked. They're not picked up on the Illumina platforms. KRAS variants still fail as design. They're very GC rich. They're hard to pick up, so people haven't been following them. So we went through incredibly painful efforts, again, in the day before technology is what it is, where sequencing was hard. And we sequence all areas, excuse me, that affect microRNAs. So microRNA promoter regions, microRNA coding sequences, and their binding sites. This project started in 2009. It was just published in 2015. It was ridiculous, so painful. But what we did, we found all these mutations. We really proved that they would screw up microRNAs. That was our bar. We sorted through, did even more bioinformatics, and came up with pools that we said this would be good to test in immune therapy, in radiation therapy. These are genes that should be involved. They're microRNAs that should be involved. And if someone walks around with a difference, we think they'll respond differently. And so I'll tell you what we found. So we've done two big studies that I'll talk about. One is in immune therapy. New wave in cancer therapy. I'm sure everyone's heard about it. We are trying to get your immune system to fight your cancer. The first drugs doing that are checkpoint inhibitors. We use them now a lot in a lot of trials with radiation. The idea is that your tumor's hiding, and if we can expose it, your immune system's just waiting to come in, and it's going to take care of it. I will say it's not true for KRAS patients because their immune system is not waiting to come in. But the problem with immune therapy is people have bad side effects. So 25%, I'll give you all checkpoint therapy. I could give it to you today, cancer or not. 25% will develop in autoimmunity, where your body turns against your liver, your brain, your heart, something, and we don't know who you are. We have no way to know who is going to have toxicity to immune therapy today. Doesn't matter on cancer type, doesn't have to do with response, because you get the same if it's in a cancer with no responders versus melanoma. So we had access to a great group of patients at UCLA, where we do lots of it. Clean cohort from Tony Ribas melanoma that had known response and toxicity, used that as a training set. And consistent with our hypothesis, we said, if we can predict toxicity in melanoma patients, it's your DNA, it's you. I don't care if you have a breast tumor, a colon tumor, a head and neck tumor, I still will be able to predict it. So if our paradigm's true, it should work, regardless of cancer type. So that's what we did. We got 100 people, other cancer types, 5 institutions. All we need is your DNA and to tell us if you've gotten only that drug and if you had toxicity. Super clean, super easy, black and white. Next, we've done a lot of work in radiation. I'll just touch on one of the studies we have since we've submitted. This paper is being resubmitted maybe today. This work has been submitted to ASCO and Astro, looking at toxicity from radiation. So it's our same problem, toxicity from radiation. 90–95% of people are just fine. 5 to 10% of people have bad side effects. I have no idea who you are. I treat you all, I'll treat you the same, to the same site, the same way. Radiation oncologists, you will find your toxic patients and you will agonize and you will go over your plan and look at your fields. They'll fail or they'll have bad side effects. You treated them the same. So who are they? And if we know ahead, it will help. So we have, I'll talk about just a cohort we've done of 5-day treatment versus 8-week treatment of prostate cancer to see if we can predict toxicity and if it's different. So first, just a quick touch on the autoimmunity from these new agents. They're called IRAEs, immune-related adverse events. They can affect any organ, as I mentioned. I view it it's an you push somebody and their immune system's like, hey, and just goes awry. And it's like, hey, liver, you look a little strange. I don't think you're part of me anymore. So it's just like autoimmunity itself. It's just a breakdown in normal immune regulation somehow. But grade 2 or above, which can be serious, develop really commonly, about 1 in 4 patients. And it really can lead to long-term toxicity. So lifelong diabetes, you can have hepatitis, thyroid is one of the milder ones. You just have to take thyroid replacement. But cardiomyopathy, heart, it happens. And if not caught, kidney failure, they can really lead to problems. So as I mentioned, our training set actually is 62 people now. And then we did our validation cohort, cut it off at grade 2 or above, included all types of toxicity as long as there's grade 2, didn't try by different cancer type or different tissue type. And what we found was a signature that applies. I could test you all today and tell you if you're going to have toxicity no matter what type of cancer you develop. So we found different biomarkers, and these are some of our top. This one in RAC1, a gene, really important. This is a microRNA binding site in this gene, really important in development of innate immunity. So I'd suspect these people that have this just have a slightly tweaked immune system. If you push them and they have this marker, over time, 95% will develop grade 2 or higher toxicity if you have them on PD-1 long enough. And what we could see in melanoma, our training set, we have accuracy of about 75%, sensitivity, specificity up there. But then in our independent cohort, any cancer patient of any type, it was even better, 90% accurate. With a negative predictive value, it's actually 99%, but I could almost 100% say you will not have toxicity if I give you this. You will not. That's not your immune wiring. And what's great or interesting is that this doesn't affect response. So you might be someone where we push you, you might respond great or you may not respond at all. This signature won't tell you. So we can manage your response if you really need the drug. So that's been a little bit of a debate, and that's more in the paper. And I'll just talk about the radiation results for a few minutes and leave time for questions. What we found was very interesting. One, we treat people with radiation. There's a lot of work saying we can follow along while we treat you, and we maybe will pick up toxicity or a response. Radiogenomics, like going on while we treat. I'll tell you, in our databases, people that have short-term toxicity, they're not the same people that have long-term toxicity. You can blow through treatment and be 100% fine, and at a year have horrible toxicity. So it's clearly, they're different genetics. They're different people that have those responses. So we saw that for sure in these panels, both 5 days of treatment or 8 weeks. Different people that have those toxicities. I think long-term toxicity matters more. We have ways to get you through short-term. Long-term, we're up a creek because we didn't know it was coming. It's immune-mediated. It's fibrosis. It's not easily fixable. Something else that was very interesting, different tissues have different toxicity. That I wasn't sure I'd see. So the bowel versus the bladder, different biomarkers predicting toxicity. Must be different cells being affected. But was really good to see, and I think how we will ultimately be able to use this in radiation is we can tell you 5 days is great for you, or no, that's not safe, you should go for 8 weeks, or 8 weeks is great, or no, that's not safe, you should go for 5 days. Very different immune, very different genetic responses because it's a different stress. 5 days of radiation are huge fractions. 8 weeks are tiny little fractions. We're pushing different buttons. And so depending who you are, what button we push matters. So we thought that was really interesting. And these are just, I'm sorry, I don't have a graphic of this, but 85%, you'll find you could do 5 days or 8 weeks, whatever makes you happy. 5% of you do not do 5 days. 10% do not do 8 weeks, but the other one's safe. So that's where we are. So I'm going to summarize. I hope you believe, or I've convinced you on some level, that there's baseline genetic differences that really change us as human beings, even though you all look the same. KRAS people all look the same. Well, that's not true exactly, but when they have cancer, I usually can tell. Anyways, like I'll know they're a KRAS patient before I test them, but they have a certain phenotype. But they really, and a lot of them are these microRNA pathways. These are the non-coding RNAs. They're important in our systemic stress response. They shape us as human beings. They manage how we respond to stress. There are, I'm sure, many others. There are non-coding RNAs. We don't even know how these microRNA mutations work, to be honest. I might find it has nothing to do with the microRNA. It's just where microRNA binds, but I'll still be okay with that. They predict how you will make a tumor, but they more importantly predict how you respond to how we treat you. And as an oncologist, in a best world, we'll figure out all the triggers. It will prevent cancer, and we'll all be out of a job, and I would be so happy. I'm not sure all the trainees would be happy, but I'd be super happy. Hopefully, and we try to do some of that through our nonprofit. We do prevention. But if anything, if you have cancer, we will give you the right treatment. We will say, no, that's not safe. This is best. This is safe, and this is most efficacious. This will most likely cure you. That's what we all want to do. And it's not because we're not trying. The bummer is, I'll lead for the scientists on a bummer. The bummer is, these are not conserved in animals. I cannot model this in a mouse. It's just these regions are not conserved, the binding, the 3' UTRs. I can do tissue culture, which is totally fake, and we do see some of the same stuff we see in tumors, but I can't really model an immune system, and it's a whole systemic response. I can't model that in tissue culture. So it's been critical for us that we've had the trials, and we're trying on the science. We have much more on the trials than we do on the science explaining it. But maybe in my lifetime, I hope. But it's been critical to go back and forth, and we've had amazing collaborators and amazing opportunities. We look forward to additional collaborations here. You guys have great clinical trials. All we need are your 2 endpoints and a tiny bit of DNA. And let's find an answer for those patients that we can someday be applied. That's what we all want. So work to be done, just some of the stuff that's going on. As I mentioned, we do have some exciting validation happening, which is the clinical side. So we have a prospective trial for KRAS-variant HPV-positive patients. For those in the clinic, you may know there was a big published phase 3 saying cetuximab didn't work for HPV-positive patients. We got that trial with David's help. We are going to show for KRAS-variant that's not true. So we're excited. That's that trial. We have prospective collections going on predicting immune toxicity in clinics, saying, use this, how is it helpful? And if you don't treat those patients, how common is toxicity? And we're finding amazing results. Clinics that went through with 40% toxicity have 5% toxicity in patients. And we're starting in the clinic incorporating this prostate decision tree with patients. Because you know what? When people are calling my chairman a year out with urethral necrosis and saying, I'm going to sue you, you didn't tell me. And he says, I did tell you, it's documented. He wants to be able to tell them ahead. Say, you're at 90% risk of toxicity. It's up to you, but you have to really want to do 5 days. It's not 100%, but it's high. So it's a joint decision. And sometimes there's not another option, but it's helpful to know. Not for the patient, it's helpful to know. And then the laboratory, I just got another R01 to look at predicting toxicity to radiation plus immune therapy. We have prospective trials doing that. We have several collaborators who have given us cohorts. We have lots of work to do in predicting radiation toxicity. There are so many applications. Preoperative sarcoma, where 5% of people never heal their wound. Breast, where people have reconstruction. Small percent have horrible radiation fibrosis and they lose their implants. It's awful. Small, rare, but we should avoid if we ever can. So great patient cohorts, great clinical collaborators who have helped identify and define these needs. We have a huge library of transformed cells for patients where we're hoping we can model some things, at least DNA repair capacity with these different genetic mutations. That's going on. And I'd love input for great immune models. Our problem, again, none of our mutations are in mice. So we can use humanized mice, but it's hard to know if it will really represent the systemic response to guide us. And that's it. And so I have a growing lab at UCLA, always open to new, excited postdocs. A lot of people we've had some great students and postdocs that have already gone on. I had a great group at Yale that really worked for years on these studies, and some good funding, and I thank you all for your attention.
Yeah.
Beautiful work. Just wondered that for cancer types that have very high KRAS mutation rate, long temporal,
Yeah.
have you look at the patient population that the KRAS mutant versus KRAS variant?
Yes, great question. So in lung, that was looked at early because we see it's a risk for lung, so it's more common in lung. It's relatively rare someone will also have a KRAS. It's not mutually exclusive, but it's pretty rare. It's in like 20% of non-KRAS mutant lung cancer patients and like 5% of people with KRAS mutations. In colon cancer, they've done a lot of work with that. And what we find, again, not mutually exclusive, but actually, even if you have a tumor KRAS mutation, if you have the born KRAS variant, they respond to cetuximab. So it actually trumps KRAS mutation in drug response, which is very interesting, and they love that in the colon cancer world. So not mutually exclusive, but a lot of the other cancers, we see it enriched in head and neck, triple-negative breast, ovarian. They're known to use this pathway, but they don't have tumor mutations in KRAS. So it's not just a surrogate for ultimately getting a KRAS mutation.
I actually had the same question, but building on that,
Yeah.
biology, how do you reconcile the KRAS mutant with the KRAS variant? Do you see a different microRNA dictating the KRAS mutation, or is there a pathway where they cross over?
Well, I do think it's actually, they're really fundamentally different because I don't think the impact on the KRAS protein itself is as important for the KRAS variant patients versus the KRAS mutant. It's a much more global impact. So, you know, we do see KRAS addicted signatures by the time they get to them, but their cetuximab acts upstream of KRAS and still can help the patient's immune system. And that doesn't work for KRAS mutant tumors. Although if they have both, again, it still helps them, prolongs their survival. So I think ours, as I said, I think it's more about like how you're built as a person, like your whole body, and it's about your systemic response to whatever stress we give you, because that's what microRNAs do, versus a single pathway within a tumor. So it's a much more global. And another example is EGFR inhibitors for mutant lung cancer, gefitinib, work great generally for EGFR mutant patients. If you have the KRAS variant, it is a horrible drug. They all die within 6 months. And so we've seen that in 2 battle trials and another trial as well. So it kind of trumps, that whole body response trumps that single tumor mutation. And I do think in oncology we're looking more at the whole body with immune therapy. That's what it's about, right? It's not just, we didn't find a lot of other driver mutations. So I think we're realizing that that isn't the answer for everything, but yeah.
So your LET7 was substantially lower.
It is significant.
So does that suggest there's a positive feedback with it? You know, what happens if you knock down the KRAS drag? Does LET7 go back up?
Yeah.
And is the KRAS variant signature enriched for LET7 targets?
That's a great question. And we do think there's a positive feedback loop. And in one of our papers, we actually used siRNA that matched the LET7 binding site, and it reversed the phenotype and decreased growth. So it is, that seems to be the tip of the iceberg, that miscommunication. And which makes sense because we knock that single spot in and we recapitulate this whole mess. We thought about trying to do like siRNA therapy. As I mentioned, though, delivering microRNAs has been incredibly like systemically toxic. So, and as far as enrichment for LET7 targets, that's a great question. We do find in the NCI-60 panel that it was consistent with that. That is what we found actually, and it's a very specific bad tumor phenotype, and I'm forgetting what it's called, that is associated with LET7, and we see that in the KRAS variant cell lines. So thanks. Good question. Anything else? Yes.
So you mentioned that that estrogen deprivation occurred at menopause when they are treated for women who have the variant.
Yes.
And do you have evidence, for instance, that estrogen deprivation drives to females who are likely to have triple-negative breast cancer versus ER positive, who be estrogen dependent or something?
Yes. Great question, and I didn't really mention that. We do. We know having the KRAS variant is a risk for premenopausal triple-negative breast cancer. And it's estrogen is protective. And in our cell line models, and I didn't show this, if you take away estrogen, serum sarmatecoestrogen from the KRAS variant versus normal epithelial breast lines, it causes transformation. They develop anchorage-independent growth. And what we've actually figured out is that they're protected through this GPER pathway. It's a G, which that's what I love and hate about science. I'm like, what the frick is a GPER pathway? Like, what the hell is that? Or metabolism? I don't know anything about that. Like, you get dragged into other areas, but it turns out if you use GPER inhibitors, it totally blocks transformation. They need something bound. And it is an inhibitory pathway that can either go through KRAS or the inhibition blocks that pathway. So they need estrogen to protect them, basically, I think, to keep it balanced and in check. So we're really actually just getting much more into that, into those studies. We just got some funding for GPR. It was just so hard. Cancer prevention, and I'll tell you, unfortunately, breast and ovarian cancer space, for genetics, it was a very contentious field with the whole BRCA thing and Myriad, and then GWAS saying they found all markers. So we come along and, oh yeah, this predicts ovarian. I mean, you think I was a child killer. I was persecuted. It was horrible. It was terrible. It was crazy. So we're like, okay, let's focus on cancer therapy for a while. We'll get back to women's health. It's in the nonprofit now. And the studies have gone on, but science is a weird place like that. There's egos you tread on. You're like, oh, yikes, that you didn't know existed. And you just have to be conscious and sensitive and only push in different doors. That's why I always tell people, you kind of push, you get through the doors you can, you gently push. If there's lots of resistance, you're okay, not ready for that, that's fine, we'll move on to this. And so we've been fortunate that way. Yes?
In the triple negative space, does the KRAS variant overlap with a particular subtype of triple negative?
That's right. So we had a paper in Lancet Oncology about this, but it looks like it's more basal-like. And we haven't gone beyond that. So that was a cohort of 150 patients from Yale where they did their whole gene profiling from their old paraffin-embedded tissue. So it did seem to kind of mesh in, but not a perfect mesh. And I know there's newer subtypes now, actually, and we just haven't matched the two.
And then you said that Xanax is out, which totally makes sense. But how well, in lung cancer or any of the other cancers, does the variant correlate with high Z expression? Then you can just stain for Z.
I know, we haven't looked. We haven't, actually, I've never looked in tumors, except we only looked in the cell lines. We just haven't. Someone may have all that data, and we would love to, correlate it. I think working with other groups is a great way to kind of confirm some of the biology that we see. So I don't know. That's a great question. Yes?
I know you said you looked largely at long-term side effects. How about radiation pneumonitis? Have you seen anything that might predict?
Yeah, so we're looking at that. And we do look at acute too. And I'll tell you, all of our markers for acute are in ATM. Totally makes sense. DNA repair versus our long-term side effects are more immune-related, like PPTN2 and stuff like that. Pneumonitis is one of those kind of in-betweens because it's clearly an inflammatory response. And I found lung just really challenging because everyone always denies it's related to their therapy. So for immune therapy, the problem is with progression of lung disease, you look like you have pneumonitis. So is it really the radiation? Is it the immune therapy? Or is it just progression of disease? So I feel like it's actually been a really challenging endpoint that's not that clean. So we have lung cohorts. I just haven't dove in yet, but I will. It's on my list to do, just not at the top because I'm not. It's too hard to, it's just been hard to clarify. Sure, if you respond, or do they have an infection because they have post-obstructive pneumonia, right? I think if they respond to steroids, it's pneumonitis, call it what it is, but it's been a little harder.
We have, let's just take one more question from Dr. Brown.
Fantastic work, Joanne. This is wonderful.
Thank you.
From my clinical perspective, can you correlate at all the viral status with toxicity? There's some work from Anderson showing that those who are HPV positive tend to have, in general, more acute mucositis, longer toxicity in terms of fibrosis, fistulas, and things like that. Can you comment on that at all?
Interesting. I haven't looked at that, although we do have a trial that we're profiling right now for toxicity. And then we have the prospective one. I don't know. Long-term.
The virus could, manipulate the tumor microenvironment in some way, maybe.
I do. I think they're very immunogenic. I think that's why they respond so well. Is once you show the body, hey, there's a virus in here, the body's like, whoa, bring the immune system in, which is a double-edged sword. So if there are people that maybe have a hyperactive immune system, I would imagine they might have worse long-term toxicity. In head and neck's a little weird because their acute toxicity, I do think, is inflammatory as well, besides just DNA damage. So I'm not sure how the markers will show up in that one, but I don't know. It's interesting. We have both the 0522 cohort as well, all the samples to look at biomarkers as well as an internal one and the prospective trial where we are going to try to look at toxicity. So, yeah, absolutely, we'll do that prospectively.
Okay. Well, thank you very much for your participation.
Thanks, everybody.
We want to thank you very much.